So it might affect folding of other proteins. That's getting into a more complicated scenario. One last.
Well, you produce an active form of RAS, then it wouldn't be able to interact with the effector molecules, and that could be, and if it's inhibitory, then it could generate an inhibitory response.
Okay, you're very close. Very nice thinking. He said, well, maybe because it can interact with effector and it has something to do with interaction with effector. In fact, it's the other side. It can still interact with the guanonucleotide exchange factor. It's just that that interaction, because of the mutation, it's unable to nucleotide exchange. But the interaction is still possible. And if you have a dead form of RAS, a non-activatable form of RAS, interacting with GEF, if you have enough, if you overexpress, the more and more you express of the inactive form, the more and more of GEF you tie up and prevent it from interacting with the normal RAS. So if I express 10 times the amount of mutant RAS over the normal level of RAS, then 10 times the amount of GEF is now interacted with in a non-functional way, and you tie up all the GEF so that it can't activate normal RAS again. So in that way, it could act in a dominant inhibitory way, and that's been one really important tool for understanding the role of what types of effectors there are for normal RAS. Okay, so now the question is, we have two oncogenes that we've identified. We've identified SRC, for which we know there's a normal cellular SRC that can be mutated to become oncogenic. We know that there's a normal cellular RAS that can be mutated to become oncogenic. And the question is, are they part of the same growth control pathway, or are there multiple growth control pathways that each one plays a distinct role in? Are there parallel acting growth control pathways, or are they part of a single common pathway? And in order to answer that, the following type of experiment was done. We have two tools in hand. We have an activating mutant form of each of these viral or oncogenic forms of the protein. So we have an oncogenic SRC, which is hyperactive or constitutively active. We have an oncogenic RAS, which is constitutively active. Okay. And we can put each one into a cell, and it can cause uncontrolled growth. So we have a nice assay for their activity, physiological assay for their activity. If we introduce an active form of one and block the activity of the other, we can now ascertain whether they are components of the same pathway, and we could also determine the ordering of those components. So for example, in the example shown here, we introduce active SRC and at the same time inhibit RAS. In this case, in the early experiments, it was done by injecting an antibody of RAS into cells to knock out its function. Another way of doing it would be the dominant inhibitory form of RAS. Okay. So we activate SRC, we block RAS, and ask whether mitogenesis is caused by SRC is blocked by blocking RAS. Or we use an activated form of RAS and block SRC, and ask whether mitogenesis through RAS is blocked when SRC is inactivated. And the results are that SRC's ability to induce mitogenesis was blocked when you blocked RAS, but RAS's ability to induce mitogenesis was unaffected by blocking SRC. So what does that mean about the relationship between these two components? Yeah. Yes, excellent. So RAS is downstream of SRC in a common signaling pathway. Okay? Because only if RAS is downstream would blocking RAS be able to block SRC action, but blocking SRC would not be able to block RAS action because RAS is downstream in the pathway. So this type of analysis is used today as well, activating, inhibiting mutations or biochemical analyses where you activate at one site, inhibit at another site, and determine whether components are part of a common pathway and the ordering of those components. Okay. So we know that this tyrosine kinase, SRC, as a tyrosine kinase is upstream of RAS. Somehow this tyrosine kinase is influencing GEF to turn RAS on, and we need to understand that. And we also need to understand what the active RAS is doing. So we have a tyrosine kinase influencing RAS function in a way that still needs to be defined. And we have RAS function somehow causing, downstream, somehow causing uncontrolled growth. And so the question is, what are the RAS effectors, right? The same way we needed to understand what the G protein effectors are. The G protein effector for adrenaline is adenyl cyclase, okay, and works through second messengers and amplification. What is the downstream effector of RAS in this growth control pathway? And the answer to that came from the similar type of approach that I've described, okay, for how RAS and SRC were initially identified. Okay. What would that approach be? How would you identify other components of this pathway? Yes? Sorry? Radioactive GTP. So you're now probing RAS itself. Okay? GTP doesn't get transferred to the effector. So how would I identify other components of the pathway? There are actually 3 components that I'll describe in a minute. But how are these components identified? How would you go about identifying them? Yeah.
By inhibiting RAS, and then doing using them in the downstream and upstream, inhibiting RAS and taking one of the other factors.
Well, the question is, what other effector? I don't have any effectors in my hand. So you're sort of describing something that I can do later, but how do I get an effector? How can I identify additional effectors in this pathway? Yeah. Knock out what gene? That's the question. Yeah.
So SRC is a tyrosine kinase, so wouldn't you want to find out what it phosphorylates?
Okay, so you want to know what's in between these, but what I'm asking is what's downstream of RAS. Okay? You're going to kick yourself if I tell you the answer. How did I discover SRC? How did I discover RAS? How will I discover additional members of this same growth control pathway? Yeah.
Do one cell with the RAS activated high level and one without, and then do a protein assay to see if there's new bands of protein or more than.
Okay, so that's an interesting approach. It starts to get at sort of what is the biochemical, what could RAS be biochemically interacting with. That's very good, and we'll get to that in a minute. One last attempt. Yeah?